All right, welcome to chapter 8. So now we'll learn a little bit about genetics, which is actually my specialty. That's what my PhD was in. So without further ado, let's get into it. So, um, okay, so microbial genetics and genetic engineering. Okay, so make sure you guys go over this. I'm sure it'll be one of the printouts. Okay. Okay, so we all heard about DNA. I'm sure you guys heard of seen the double helix before, right? So DNA is a molecule, which is a type of nucleic acid. So all cells use DNA to store genetic information. Okay, which is what the cells need to produce proteins. Okay, so genetic information is your DNA. Okay, and inside you, your DNA tells the cell when and how to produce certain proteins at certain times, which is what makes you who you are. Okay. Okay. All DNA is composed of nucleotides. There are three parts of nucleotide. There is a phosphate group. Okay. There is a sugar, and there's a nitrogenous base. Okay. There are four possible nitrogenous bases in DNA. Okay, adenine, guanine, cytosine, and thymine. Okay, or A, G, C, and T. Okay, these covalent bonds join together nucleotides to form DNA strands. Okay, the um, the two sides of the DNA are the two strands, and together they form the double helix. Okay, the strands run in opposite directions to each other. Okay, so you see it one strand running this way, one strand running the other way. Okay. Notice the P's are right side up on this side, the P's upside down on that side. Okay. So they run anti-parallel to each other. Okay. And if you look at them, the A's always bind with T's, the C's always bind with G's. Okay. This is known as Chargaff's rule. So C's always connect to G's, G's always connect to C's, T's and A's, etc., etc. Okay, so this actually makes it really easy. And once Watson figured this out, um, it became really obvious how DNA replicated. Because if you have half the ladder, okay, you can easily understand what the other half should be. Okay, so one strand is called the template strand. The other strand is called the anti-template strand or the complementary strand. Okay, so A's bind with T's, okay, C's bind with G's, okay. So this is really easy. So you just page. So here is A, T, G, G, C, A, C, G, T, A, etc., etc., etc. Okay. Why does DNA replicate? Because you need to copy your DNA into new cells. Okay. All right. Okay. Um, so the DNA packs tightly into chromosomes. Okay. Uh, so the DNA, the organism's genome is packaged DNA into cells. Our DNA isn't naked. Okay. So this right here will be what's called naked DNA. Our DNA is usually wrapped around in proteins called histones. Okay, so it usually wraps like this. And when we want to actually move the DNA around, like for cell division, we actually even have to pack it up even tighter into chromosomes. Okay. Okay, the chromosome is a distinct cellular structure composed of a neatly packaged DNA molecule. Eukaryotic chromosomes are found around histones. They're located in the nucleus, okay, and they're usually found in pairs, so diploid or occasionally haploid, and they're linear in appearance, okay, and this makes them very different than bacterial chromosomes. Bacterial chromosomes are circular, okay, and usually they only have one circular chromosome, okay. Okay. The genome is the sum total of genetic material of the organism, so all your genes, okay? Most of the genome exists in the form of chromosomes, okay? Um, but not all of it, okay? Some appear as plasmids or in certain organelles. Um, in mitochondria and chloroplasts, have their own DNA, okay? And remember I said the bacterial chromosome is circular? The mitochondrial chloroplast DNA are circular, which is one of the reasons that we know that mitochondria and chloroplasts were once free living bacteria that we kidnapped. Okay, the genome of cells is composed entirely of DNA. The genome of viruses contains either DNA or RNA, and we already talked about this. For example, COVID-19, the SARS-CoV-2 virus is actually made out of RNA. Okay. So here you can see our chromosomes are mostly packed in the nucleus, but chloroplasts and mitochondria have their own small little genomes as well. Okay, and then viruses, and then here's a bacterial chromosome, and then the plasmids. Okay. All right. So DNA replication. Okay. So here's DNA replication. Remember I said these strands were anti-parallel. Okay. Okay. So this is what happens. So there's DNA replication. Number one, the enzyme topoisomerase works. Topoisomerase unwinds the DNA. It unzips the DNA. Okay. The helicase then breaks the hydrogen bonds, okay, and then the single strand binding proteins. So once DNA likes to hybridize, so once you unzip with the helicase, okay, the single strand binding proteins attached to it and keep them separate for a while, okay. Along comes this enzyme called DNA polymerase 3. DNA polymerase, so makes polymers of DNA, okay, attaches to the two sides, okay, and makes copies. So remember, A binds to T, G binds to C, and so on. Okay, but as the DNA unwinds, there's one strand that can go all the way through very easily, and that strand is the leading strand. The other strand is called the lagging strand, and because the DNA is facing the wrong way, it has to make short little clips of itself and then kind of shift to a new location, make a short little clip of itself, and then shift to a new location. Okay, this strand is called the lagging strand. These little fragments of DNA that are being made are called Okazaki fragments. Okay, and they are attached together by this enzyme called DNA ligase. DNA ligase ligates pieces of DNA together. Okay. Okay. So bacteria have it easy. Bacteria just have one chromosome, so it's generally pretty easy to replicate. They start from one replication fork, and they make a copy of it, and then all that they do is cut themselves out of it. Because when you do this, you form like two little links that are linked together, like links on a chain. The only thing you need to do is cut one of them and seal, like move them aside and seal it. And then now you have two daughters of molecules of DNA, which are exactly identical. Okay. Genetics is the study of inheritance or heredity of living things. Okay. It explains how biological properties transmit from parents to offspring. Okay. How these traits are expressed, the structure and functioning of the genetic material, and how this material changes over time and through generations. Okay. Okay. The three categories of genes: structural genes code for proteins. Okay. Genes that code for RNA machinery are used in protein production. Okay, the ribosomal RNA genes, and then the regulatory genes, the ones that control gene expression. Okay, genotype is the sum of all genes. Okay, is what your genes are made of. Okay, phenotype is what you actually look like, what your traits actually are. Okay. So while these two might be related, they're not absolutely identical to each other, and we can get into more details about that. Okay. In cells, genetic information from DNA to RNA to protein. Okay. So if you think about it this way, DNA is your library of information, and it's very, very important stuff, right? So you don't want it to ever leave the nucleus where it could possibly get damaged. So what do you do with that information? Because you still need it, you still need to take it to the outside, okay? So you copy the information, okay? That copying is called transcription. You transcribe something. You're literally copying it over word for word or letter by letter. So it goes from language of nucleotides to the language of nucleotides, so DNA to RNA. RNA is flexible and portable, so you can actually move it out of the nucleus very easily. And from the nucleus, you can translate it into protein very easily. Okay, so that process is called translation. Translation, you're going from the language of nucleotides to the language of protein, the amino acid sequence. Okay. The transcription is master code of DNA used to synthesize an RNA molecule. Translation transcribes RNA to produce protein. There are exceptions to this pattern. RNA viruses convert RNA to RNA. Okay, so the COVID-19 virus converting RNA to RNA. There's no such thing as a DNA sequence of the COVID-19 virus. Okay, you'll probably see that in some literature, but really they're talking about the RNA. There's no actual DNA in the virus. Okay, retroviruses. Retro means to go backwards. Retroviruses convert RNA to DNA. Okay. Okay. So RNA is a nucleic acid that actually participates in protein synthesis. Okay. Okay. So the differences between RNA and DNA. DNA is deoxyribonucleic acid, so it has the sugar deoxyribose. Okay, there's an H here and there's an OH there. Okay, so deoxyribose ribose. I know it doesn't look like a big difference, but it's a huge difference. Okay, this makes DNA super stable, and so DNA doesn't degrade very easily. RNA is much less stable because of that OH. but that OH actually allows the RNA to be more flexible and do different things that DNA cannot. Okay, so the next thing is the bases. In bases, you have A, C's, G's, and T's. In RNA, you have A's, you have C's, you have G's, but you do not have a T. The T has been replaced by a uracil. Okay, DNA is double-stranded. RNA is usually single-stranded. Okay, the function of the DNA has just storage function. Okay, RNA has a lot more functions than that. It carries the protein encoded information. It actually helps in the protein making process. It catalyzes some reactions by itself. Okay. All right. There are 3 types of RNA that you have to know. Okay, messenger RNA. Messenger RNA or mRNA carries the information that specifies a particular protein. Okay, it is the copy of the message in DNA. Transfer RNA molecules carry amino acids, okay, to the right spot. Ribosomal RNA is part of the ribosome. The ribosome, we talk about it being one thing, but in reality, there are multiple parts to it. Most of the parts are actually made of ribosomal RNA. Okay. Transcription consists of 3 parts. Initiation, how to start. Okay. So any process really consists of these 3 parts. How to start, how to continue, and how to stop. Okay. Initiation, how to start. Elongation, how to continue from the initiation, and then you keep repeating until you reach the end, and that's termination. How do you stop? Okay, so here are the 3 RNAs, and you can see what they do. Okay, the ribosome, so prokaryotes and eukaryotes are different in size. The ribosomes are found in bacteria, mitochondria, and chloroplasts, or 70S. Remember, the bacteria are primitive bacteria. This is why they all have 70S ribosomes. Okay. The small subunit binds to the 5' end of the mRNA. The large subunit supplies the enzymes for making peptide bonds. So we'll talk about what that means. Okay. Okay. So the players in translation. So this is a small subunit. The small subunit will bind to the ribosomal. The small subunit will bind to the messenger RNA. Okay. And then here you bring in the players. There are 3 sites here, EPA, the easy way to remember this, Environmental Protection Agency, exit, okay, P site, A site, okay. Okay, what happens is you bind the tRNAs to the right spot, and then what happens is the sites shift over one. The P gets added to the A, and then the A shifts into this position, and then you add the next one. Okay? All right, we'll go over that again. Don't worry about that. Okay. The master genetic code, okay, is a codon. A codon is a group of 3 nucleotides that dictate which amino acid is added to the growing peptide chain, okay? Because there are 4 nucleotides, or 4 different nucleotides, okay? A, U, G, and C. That means there are 4 to the 3rd different codes, so 64 different codes. And it codes for 20 different amino acids, which means you have more than one way to code for a particular amino acid, which means there's a lot of redundancy. Okay, built into the genetic code. Okay, so a lot of times when you have mutations, the mutation might be obvious in the nucleotides, but they're not obvious in the proteins because they can encode for the same proteins. Okay. All right, one of the things you should learn is how to use this map. Okay, this is the genetic code information. Okay, so any 3-letter code you can look up. So for example, the code U is right here. So you just look for the first base. Okay, then we'll make up, we'll write it out here. We still randomly make something up, CAC. Okay, so we'll look for the first C. So the first C is here. Okay, the second C, or second letter's A. So second letter's A. So I know I'm somewhere here. And then C, CAC. So I know CAC codes for histidine. Okay. So I'm gonna go step back and talk about redundancy. So you can see like UCU, UCC, UCA, UCG, all code for serine. Okay. CCU, CCC, etc., all code for proline. Like, especially the third letter. Usually, if you switch that, they usually end up coding for either the very same thing or a very similar amino acid. Okay, so there's a lot of redundancy built into the DNA or into the RNA code, which is the DNA code. Okay. So interpreting the DNA code. Okay. So you have this strand of DNA, it's called the coding strand. The coding strand makes a mRNA. Okay. So the mRNA is made from the coding strand. Okay. So the mRNA is actually attached to the template strand, and it copies the template strand, but that actually makes it equal to the coding strand, except all the T's are now U's. Okay. You go back to this, you look for the very first start codon. So you look for the very first AUG. So you search around the DNA message for the very first AUG, and you start right there. And so the AUG, and then you look for the next letter, the next letter, the next letter, etc. Okay. So initiation, you look for the very first AUG, and then you elongate by adding more nucleotides, more codons. Okay. Okay. You stop, okay, when you run into one of these 3 things. Okay, one of the 3 stop signs. UAA, UAG, UGA. At that point, you stop. Okay, no matter where you are in the message, if you run into a stop codon, you stop. Okay, which of the following RNA types contains hairpin loops involved in carrying amino acids? That is the transfer RNA. So these folds are called hairpin loops. Okay, because they look vaguely like hairpins. Okay. The DNA of microorganisms made out of subunits called nucleotides. Okay. All right. We will stop right here, and we'll pick it up for part 2 next.